Hello, welcome to everybody. This is the second webinar of this series on mitochondrial disorders. And I'm very happy to introduce my colleague and friend, Professor Patrick Yu-Wai-Man. He's a worldwide recognized neuro-ophthalmologist and is working both in Cambridge and in London. He's particularly focused on hereditary optic neuropathies. And today he will talk about therapies for inherited optic neuropathies, an update. Please, Patrick.
Thank you very much. Thanks for the opportunity to present at this webinar series. As Kara mentioned, I'm a neuro-ophthalmologist by training and very much interested in the field of inherited optic neuropathies. As someone working in rare diseases, I have the opportunity to be involved in a number of clinical trials, and these are my financial disclosures, but I don't own any stocks or shares in any of the companies that would be mentioned in this presentation. Inherited optic neuropathies affect about 1 in 10,000 of the general population, and the two most common groups are Leber's optic neuropathy, also known as LON or LHON, and autosomal dominant optic atrophy, also known as DOA. The unfortunate thing about these conditions is that they are progressive and result in severe visual impairment, and currently treatment options remain limited. And that's the reason why there has been so much effort to try and move things towards the translational arena. I'm not going to go over the molecular genetics of these inherited optic neuropathies, but certainly over the years, there's been an explosion in terms of the number of genetic variants that have been known to cause inherited optic atrophy. There are more than 50 mitochondrial DNA variants and more than 40 nuclear encoded genes that now have been confirmed to cause this group of mitochondrial pathology. And all of them directly or indirectly will impair mitochondrial function. I'm going to focus on Leber's optic neuropathy now. This condition is known after Theodor Leber, who was a German ophthalmologist, worked at Heidelberg University Hospital, and he's very much viewed as being the father of modern European ophthalmology, and there are quite a few other conditions named after him. We have to go back to 1871 when he published his seminal monograph describing for the first time some of the key features of Leber's optic neuropathy, namely the fact that it tends to affect young men, that it tends to have a poor visual prognosis, and it is transmitted strictly down the maternal line. We had to wait until 1988 for the confirmation that Leber's optic neuropathy was caused, at least in some families, by a pathogenic variant in the mitochondrial genome, in this case, the famous 11.778 mutation. And this seminal work was published by Doug Wallace and his colleague Eva Nikoskilainen. LON is an important cause of mitochondrial blindness. It affects about 1 in 30,000 people in the United Kingdom. And as mentioned already, this is caused by pathogenic variants in the mitochondrial genome. In particular, 3 of these variants account for about 90% of cases. They are located at positions 3460, 11.778, and 14.44. And by far, 11.778 is the most common pathogenic variant. In the UK, about 6% of cases are caused by 11.778, and roughly around 15% are caused by 3460 and 14.44. And for the remaining 10%, if you feel that this is a clear cut of Leber's optic neuropathy, at least based on the clinical features, then if it's not already been done, one should also screen for the remainder of the mitochondrial genome, because you can identify some rare pathogenic mitochondrial DNA variants. In terms of the typical presentation, this is a pretty dramatic presentation in our field, because these are people who are otherwise fit and well. They tend to be young men between the ages of 50 and 35, and suddenly they will present with painless visual loss in one eye or in both eyes, which we will discuss a bit more. And as mentioned already, there's a very rapid deterioration of vision to 660 or worse, and in about 25% of cases, both eyes are affected at the same time. If one eye is affected initially, the second eye usually becomes involved within 3 to 6 months. So it is a bilateral sequential optic neuropathy. And if you have someone presented with a unilateral optic nerve problem and the other eye does not become affected within a year, then you have to seriously consider whether this is Leber's optic neuropathy. You can get spontaneous visual recovery. It is much more likely within the first year and much more likely with the 1444 mutation. But unfortunately, this is a disease in which visual recovery, even if it does occur, is usually incomplete, and most patients will remain within the legal criteria for blind registration, at least in the UK. And this has led on to a significant amount of effort from various groups worldwide, including involvement from industry, to try and look for ways of preserving or protecting retinal ganglion cells and improving the visual prognosis. So let's talk about one of the drugs that a lot of you will have come across, which is idebenone, which is a synthetic ubiquinone analog. So if you look at this cartoon of the mitochondrial inner membrane, you can see the 5 complexes lined up, complex 1 to complex 5. And normally what happens, you have these ubiquinone molecules which transfer electrons from the complexes, eventually converting ADP to ATP. And in terms of the pathophysiology of Leber's optic neuropathy, you get an increased level of these toxic reactive oxygen species, which can be detrimental to cell survival. And then you can also get a concurrent reduction in the amount of ATP being produced. So these two mechanisms, to some extent, are contributory to the loss of retinal ganglion cells. And if you treat, for example, fibroblast with idebenone, you observe 2 things. One is you get an improvement in the amount of ATP being produced, and secondly, you get a reduction in the level of these toxic reactive oxygen species. And given these effects, this led to the launch of various clinical studies, including a randomized controlled trial for Leber's optic neuropathy, which with the results being published in 2011 in Brain. So this was a short-term study where people were randomized either to idebenone at a dose of 300 milligrams 3 times a day or to placebo. And after 6 months, the treatment was stopped and the visual outcome was determined. I'm not going to go over the fine details, but essentially, the primary endpoint was not reached. But when you look at the graph here with the idebenone group in blue and the placebo group in gray, you could see that there was an interesting trend towards the idebenone treated group doing better, with about a third of these patients responding within 6 months of treatment duration. But as mentioned already, this was quite a short period of treatment and probably was not long enough to be able to reach a definitive conclusion about the therapeutic benefit of idebenone in Leber's optic neuropathy. After the RODOS study was completed, there was an expanded access program which was launched to try and look at further confirmatory data as to whether or not idebenone was changing the natural history of the disease. And the results were published, and this is probably the most important graph where you see the proportion of eyes or patients who were getting the so-called clinically relevant recovery, which meant either an improvement of vision from off chart to at least 5 letters on chart, or if the patient was on chart to start off with, there was an improvement of at least 10 letters. And you can see here that at 24 months, about 44% of eyes had a clinically relevant recovery. But after 2 years of treatment, the benefit kind of plateaued, really. So after the expanded access program, I'm going to mention the result of the LEROS study, which have only recently been published. So in this case here, we had quite a big cohort of patients, around 200, who were treated with idebenone at a dose of 300 milligrams 3 times a day. And then they were compared with a mostly retrospective natural history control group. You can read the report in Cell Reporters Medicine to get a more detailed view of the analysis that were conducted. But suffice to say that the data was confirmatory of what was seen in RODOS and the expanded access program. So if you look at the group of patients who were within 1 year of disease onset, the so-called subacute and dynamic group, you can see here the primary endpoint being showed both for 12 months and for 24 months in terms of the clinically relevant benefit. And what the data shows here is that there is a positive impact on visual acuity outcomes at both 12 and 24 months. If you look at the chronic group, which has always been a subject of debate before the LEROS study was published, you can see here again that there is a significant difference between the idebenone treated group and the natural history cohort, suggesting again that idebenone was worth using for patients who were between 1 and 5 years of disease duration. Obviously, we need to take that information context, not everyone treated with idebenone will respond. And what I will show you again here was there was a rather quirky, a result from the LEROS study, suggesting that the 3460 mutation could have a detrimental effect, but it's not clear whether this is a real effect or just an issue with the cohort and with the analysis of a small group of patients. So if we look at the 3460 subgroup, you can see here that the numbers start to go down quite a lot. We had 26 patients in the idebenone group and 18 patients in the natural history group. And you could see here that essentially there was more clinically relevant recovery in the placebo group compared with the idebenone treated group, which was not significant. But if you look at the data for what we term as being clinically relevant worsening, you can see here that there was a significant difference with the idebenone treated group doing significantly worse. When we looked at the chronic group, this was not observed. So you can see here that there was nothing significant showing in terms of these two parameters for the chronic group. So what do we take from here? I think essentially it seems that the 3460 mutation is not the best in terms of response to idebenone, especially in the chronic stage. In the acute stage, it is a bit unclear, again, as mentioned already, whether we're dealing with a real effect or whether there are confounding variables here. To reinforce the fact that in RODOS and the expanded access program, a detrimental effect of idebenone in the 3460 group was not observed. Having said that, we contacted some experts, both in North America and in Europe, to try and get an idea about whether or not they are using idebenone for patients with 3460 mutation. So in North America, it's about 50-50%, whereas in Europe, most clinicians that were contacted were using idebenone for the 3460 mutation. We need to do more work on this and see whether or not there could be other nuclear genetic variables that might be influencing response or perhaps a detrimental effect, for example, NQO1. But for the time being, I think we need to basically take the data as it is. The other thing that we have not yet clarified is whether or not the timing of treatment is relevant, because there is some data suggesting that treating too early within 6 months might also not be as good as treating later, for example, between 6 and 12 months. And then we also need to look at the group of LOND cases where they present very early, before the age of 12 years old, because childhood LOND tend to do better in terms of the visual prognosis, but there are certainly clinicians that decide to treat children when they are 12 years or younger. In terms of where you can get access to idebenone, well, the Raxone formulation was approved by the European Medicines Agency back in 2015. And at this point in time, there are roughly around 16 countries where there's a national reimbursement program, and there are 15 other countries where you can get access to Raxone on a named patient basis. So this is not perfect, but at least we have an approved treatment, and all the data so far from RODOS to the EAP to LEROS confirm that there is a treatment benefit from using idebenone in the context of Leber's optic neuropathy, both in the acute and the chronic stage. So we need to look into other treatment options, and the obvious treatment option is gene therapy. Why don't we simply replace the defective mitochondrial gene? The problem here, without going to the more complex reasons, is that there is a problem in terms of penetrating the mitochondrial compartment and inserting a new genetic material into the mitochondrial compartment. So what some people have decided to do is to make use of allotopic gene expression, where instead of inserting the gene directly into the mitochondrial genome, you use a roundabout way to doing this. And this was shown quite nicely by the group of Eric Sean and Giovanni Manfredi back in 2002, so many years back, for the ATP6 mutation. So what do we mean by allotopic gene expression? So if we don't take the direct route, we could insert a recoded version of the mitochondrial gene of interest, in this case ND4 for 11,778, into the nuclear genome. It produces the mRNA, and then the mRNA gets converted into the protein, and then it has a specific mitochondrial targeting sequence, so-called MTS sequence, which allows the mRNA to be channeled towards the mitochondrial surface and the protein to be incorporated into the mitochondrial compartment. Using this technique of allotopic gene expression, the group of Marisol Coran-Debrinsky and José Sael in Paris and the group of John Guy in Bascom Palmer in the US have shown that you can rescue retinal ganglion cells with preservation of visual function. So this led on to the development of these AAV2 viral vectors to protect retinal ganglion cells against the LON 11.7 mutation. And this is just a cartoon here showing the ND4 gene construct within the AAV2 viral vector. And the good thing that we can do as ophthalmologists is that we can inject things directly into the eye, and therefore there's no need for more invasive surgical procedures. So this is just a summary slide, really, which I think is quite, to some extent, a pragmatic view about how difficult it is to do translational research, because over a period of 15 years, we've moved from preclinical work to launching these phase 3 clinical trials, and that's the reason why we need to make it clear to the patient community that translational research takes a lot of time, and the reason why it takes a lot of time is because we need to be sure about safety and we need to collect long-term clinical data. We were very fortunate to put together a group of investigators from North America, Europe, and going as far as Taipei in Taiwan. And the reason why you have to do this is because LON is still a relatively rare disorder, and to be able to recruit reasonably quickly, we need to extend our network quite widely. So this is just a quick summary of the first phase 3 clinical trials that was launched in collaboration with GenSci Biologics. You had the rescue study where participants had visual loss within 6 months. And then you had the reverse study where participants were between 6 and 12 months. So one eye was injected with the gene therapy vector, whereas the other eye was a sham injection. By sham, we mean that the eye was pressed hard with cannula, but nothing was injected inside the eye. 39 patients were recruited into rescue, 37 patients were recruited into reverse, and this is basically just signposting where the papers have been published. The reverse study was published in Science Translational Medicine in December 2020, and the rescue study was published in Ophthalmology in 2021. Now we have the benefit of 5-year long-term data, and this is just a graphical representation of these data points. So in red, you have the cumulative groups for rescue, reverse, and the long-term restore study. And then as for idebenone, we were not able to get sufficient prospective data, so we had to compare this to a mostly retrospective natural history data set. Overall, over this long-term follow-up period, you had a significant difference between the treated and the untreated group of more than 15 ETDRS letters. Last year, the REFLECT study was published. I won't go into it in any great detail, but REFLECT was different in the sense that in REFLECT, you had people being treated with the gene therapy vector in both eyes. And there was a suggestion that essentially bilateral treatment was better. And if this is the case, it adds weight to the fact that there is a biological effect, because the more you give, the better the response. The other important thing that has come out from these long-term safety studies, now that we have more than 200 patients that have been treated with the GS10 Lumevoq treatment, there's no serious ocular adverse events. You can get a bit of inflammation, you can get transient intraocular pressure rise, but most of these can be treated either conservatively or with topical treatment. And what came out of REFLECT was the fact that the safety profile for patients in the bilateral treatment group was as good as the unilateral group. So there was not any major concerns about an increased rate of complication by giving the gene therapy vector in both eyes. So moving on to the where we go from here. So obviously the results were promising and kind of generated a lot of publicity, but we also need to be very careful that there are a lot of unanswered questions and we need to get more data, because as it stands at this point in time, this GS10 Lumevoq gene therapy treatment is not available, has not yet been approved by the regulators. So where do we go from here? I think it was quite clear that we are not able to stop this catastrophic loss of vision in the acute stage. And one thing that you could put forward is whether or not you've already missed the boat if you only start treatment after the onset of visual loss. So if you look at the graph here, which shows the change in logMAR visual acuity on the y-axis against the time since onset of vision loss. So if the graph is going down, vision is getting worse. You can see here that both for rescue and reverse, we are not able to prevent this very rapid loss of vision in the subacute stage. So the conclusion we draw from here is that replacing the ND4 subunit by itself is not able to prevent the catastrophic loss of retinal ganglion cells. Why that is the case, we don't know. Probably there's some kind of refractory period, whether you're treating with idebenol or with gene therapy, and we need to look into this further because clearly this is going to limit the benefit of any treatment. And again, this is just an observation. As for idebenol, there is some data emerging that potentially treating too early might not be the best option, and actually waiting 6 to 12 months might result in slightly better vision. And again, these are observations, and we need to look into matters further. The other thing that is a matter of great debate is whether importing the ND4 subunit into the mitochondrial compartment actually allows this big subunit to be inserted and to assemble into the large complex one microstructure. The other question as well is, if you're injecting the gene therapy vector in the intravitreal space, you're hoping that the retinal ganglion cells will be transduced. But again, there's limited data about how good this intervention is in terms of transducing the retinal ganglion cells within the central retina, which is really what is relevant to us in terms of improving the central vision. And the other thing that came out, which was really puzzling and basically led to a lot of controversy, is the fact that, remember that for these gene therapy program, one eye was injected with gene therapy vector, but the other eye was a sham treatment. But when we look at the results, both for rescue and reverse, you saw this parallel improvement over time, both for the treated and the untreated eye. The first thing to say is that other groups have found the same thing. So the late Dr. John Guy and Pascal Palmer reported the same bilateral improvement. And we also had these observations coming out from the Chinese group of Professor Li Bin in Wuhan. To be able to understand this a bit better, we went on to look at these non-human primate studies where essentially one eye was injected with the gene therapy vector, GS10, whereas the other eye did not receive any intravitreal injection of the active vector. And then the animals were sacrificed, and then various analyses were done both in terms of detecting and quantifying the viral vector DNA in both eyes and various parts of the brain. And to cut a long story short, right, there was evidence of transfer of viral vector DNA. So without any big surprise, you were able to detect the viral vector DNA on the same side, up to the primary visual cortex. But when you look on the contralateral side, you could also detect the viral vector DNA in the contralateral non-injected optic nerve and retina and the optic tract up to the lateral genicular nucleus. So that possibly provides an explanation about why you're getting this unexpected bilateral improvement. But this is not the end of the story, because all of this is speculations. We're speculating that there is transneural spread of the adenoviral vector. We speculate that possibly there could be astrocytes involved in the trafficking of these vesicles containing mitochondria. And there's also the other possible explanation that this is not just the viral vector in itself, but by giving an injection in one eye, you're triggering some kind of subclinical inflammation, some kind of inactivation of the unfolded protein response, and that in itself is producing mediators which are going to the other eye and resulting in improvement of mitochondrial biogenesis. So all of these things are big unknowns, and we need further work looking into this. To add to the data here, this was presented by Professor Alfredo Seidel at the NANOS meeting last March, where we were able to get access to the eyes of a patient with LON who was treated as part of the gene therapy program. The patient died of acute alcohol toxicity, so not something that was directly related to the treatment, and then analyses were made in various parts of the eye and also various parts of the visual system. So this is just here the summary of the work that has been done so far, where if you look at the ND4 transgene, you're able to detect it in both the injected and the uninjected eye, but the level in the uninjected eye is about 100 times, 1/100, what you could find in injected eye. So could such a small level of the gene therapy vector going to the other eye cause the improvement? That is still a matter of debate. But clearly, when we designed Rescue, Reverse, and Reflect, and we were using the other eye as an internal control, now for future studies, we cannot do this, because if there is a bilateral response, we need a control group where no treatment has been given to be able to do some comparative analysis. So there are further studies coming around the corner. Biologics is planning the RECOVER study in 2025, where this will be a study where patients will be randomized either to a bilateral injection of the gene therapy vector or to placebo injection of balanced cell solution. So they're getting an injection, but it's an injection of just a balanced cell solution that we use for cataract surgery, for example. And they will be recruiting patients with disease duration between 6 and 18 months. And the reason why they've gone for this subgroup is because they've analyzed the data and found that this group of patients were more likely benefit from the treatment. The other thing that we also need to look into is whether or not treating the chronic phase of the disease could be relevant for gene therapy. Because if you go back to idebenode, what we saw from Leros is that treating between 1 and 5 years still resulted in a benefit in terms of treatment response. So in collaboration with the National Institute for Health Research, we are currently in the process of getting regulatory approval to treat a group of patients for between 18 months and 5 years. They will be randomized to either bilateral injection or to a sham treatment. So this will be sham rather than placebo. There are other drugs which are in the pipeline. Not all of them will reach the clinical arena, but just to tell you that this is a very active area of research. The other thing that we need to consider is whether or not we should try and correct the underlying genetic defect before the problem has started. Because if we're not able to prevent that catastrophic loss of vision, we need to look for a different strategy, right? You will have heard of CRISPR. People have won the Nobel Prize for this in 2020, but you cannot use classical CRISPR for the mitochondrial genome because it has a different evolutionary structure. But now people have come up with ways of doing these genome editing, these base editing in the mitochondrial genome. People in Cambridge, like Michal Minchuk and others in North America. But now the key question is, how do we deliver these base editors, efficiently, safely into the eye? How do we prevent off-target effects? And then whether or not this is going to prevent the onset of visual loss. Of course, the other thing that we need to consider is whether or not, even at this point in time, we can prevent the maternal transmission of these pathogenic mitochondrial DNA variants, because a lot of lung carriers will be women who themselves will not become affected, but who are at an age where they can still have children, and they might want to prevent the transmission of their mitochondrial DNA variant. So there is this mitochondrial replacement therapy program. The law had to be changed in the UK back in 2015. There's also a similar program which is ongoing now in Australia, and women and children are probably have probably been born already, but there's not been any confirmation of what the data is. So we need to watch this space. So this is my first part on Leber's optic neuropathy. I'm just going to move on to autosomal dominant optic atrophy now. This is a disorder which affects 1 in 25,000 people in the UK. There is progressive loss of vision from early childhood. Again, as for Leber's optic neuropathy, we have the preferential loss of retinal ganglion cells. And the main gene is OPA1, which accounts for about 60 to 70% of cases. As some of you will be aware, mitochondria are not small jelly beans sitting in the cell. They exist as a highly interconnected network with constant fusion and fission. And it has a very important role. Mitofusin 2 mediates outer membrane fusion, whereas OPA1 coordinates inner membrane fusion. So OPA1 as an inner membrane protein will be involved in a number of functions, including the maintenance of mitochondrial fusion, but also very important in terms of the regulation of mitochondrial ATP production and also the control of apoptosis. So going back straight into treatment, well, idebenone has also been used in the context of OPA1 DOA, and the Bologna group, of which Chiara is a member of, has published some case series, retrospective case series, suggesting that idebenone can improve or stabilize vision. I think the next step here is really about conducting a larger, properly powered randomized controlled trial to be able to answer whether or not idebenone has a place in the treatment of patients with OPA1 DOA. One of the problems that we're faced with in the field of inherited optic neuropathies is that we do not have easy access to diseased retina and diseased optic nerve. And that's the reason why we and others have gone down the route of producing these iPSCs from, for example, skin biopsies donated by patients with various mutational subgroups. And this is just a very quick summary of the work that we have been doing in collaboration with my friend and colleague, Professor Michael Cheetham at the Institute of Ophthalmology in London. And these are the various people that have contributed to the work that we have published, Paul Sladen, Josh Harvey, and Michael Whitehead. And this is essentially the list of OPA1 iPSCs that we have been working on. And we've optimized the protocol for producing retinal ganglion cells. You can see a very nice picture of a soma and the axon projecting out. It looks like a retinal ganglion cell. And of course, we basically need to do some further work in terms of characterizing the functional activity of these retinal ganglion cells. So one thing we wanted to do is whether or not correcting the underlying OP1 variant could improve mitochondrial function. And the results were very promising. So this is just a little summary here. The data has been published already in 2021, but you can see that in OPA1, you get a problem with mitochondrial respiration. You get a reduction in the amount of ATP which is being produced. But when you correct the underlying genetic defect in the cell lines, you are able to improve the level of mitochondrial respiration, and you're also able to improve the amount of ATP being produced. And in support of this functional improvement, when you look at the mitochondrial network, you're also getting a rescue of the mitochondrial network with less fragmentation because of the profusional ability of OPA1. How will that work out? Well, again, as for mitochondrial gene editing, this is something that needs to be looked into much more closely in terms of the safety and the efficacy of such an intervention. What other therapies are coming around the corner? Most of these genetic variants will cause haploinsufficiency, meaning that you get a reduction in the amount of wild-type protein to about 50%. So in theory, if you're able to introduce the gene again, if you're able to increase the level of the wild-type protein, you should be able to improve the survival of these retinal ganglion cells, and you should be able to improve the visual outcome. So a number of therapies are being looked into. These are slides provided to me kindly by Guy Lenaire from Angers, where they're working on mRNA transplicing. So this is just a cartoon here of the mRNA for OPA1, showing that there's a clustering of these OPA1 variants towards the terminus. And essentially, if you have a mutant mRNA which is being produced, most of them will be degraded by nonsense-mediated decay, and therefore you end up with a reduction in the amount of OPA1. So if you're able to introduce a transplice here, so it's just essentially a kind of switch around, right? Then you're able to correct and remove the OPA1 genetic defect, and this will then lead on to the production of wild-type functional OPA1 protein. And the advantage of this method is that you cannot have overexpression of OPA1. So this is just early data in OPA1 fibroblasts showing that by using the transplicing method, you're able to improve the level of OPA1, which is the first step. And then when you look at some of the downstream consequences, as mentioned already, you get a lot of fragmentation in OPA1 cells, but by using the transplicing strategy, you're able to restore mitochondrial fusion. So these are all very promising preclinical data. The other thing which is being looked into is the use of these antisense oligonucleotides, where you're using these ASOs to improve the efficiency of translation. So what you can see here is that normally you produce a proportion of mRNAs which are non-productive, and these will get degraded. But using these antisense oligonucleotides, you're able to modulate mRNA splicing, and by doing this, you get much more of the functional mRNA being produced, and you increase the level of the wild-type OP1 protein. So there are a number of phase 1 clinical trials being launched, one by Stoke Therapeutics and the other one by PYC Therapeutics. So Osprey, which is the one for Stoke, the first in human study. This is being conducted in the UK with various collaborators, including Marcel Vetruba, and we hope that the first patient can be recruited in October, so very soon. The Sundew is a study which is being conducted, at least initially, in Sydney. And again, this is a phase 1 study looking at an ASO construct for patients with OP1 DOA. As you would have realized, developing program for a specific gene for a specific variant is something that will take time and will be very expensive. So is there a way of developing mutation-independent gene therapy which would work for a number of genes and a number of variants? So people have been looking at these neurotrophic factors because these neurotrophic factors can enhance neuronal survival. And this is irrespective of the underlying gene and irrespective of the underlying disease mechanism. So a number of neurotrophic factors have been looked into. This is the list here. I'm not going to go into them in great detail. The good thing about neurotrophic factors is that they can diffuse away from the cell in which they have been secreted. So this has obvious advantages for the central nervous system. And this can be used irrespective of the underlying gene or the underlying disease mechanisms. And the reason why I mention this is because these alternative therapies are relevant not only for inherited optic neuropathies, but for other acquired optic neuropathies. And the big one out there is glaucoma, where some people will progress despite maximal medical or surgical treatment. So this is all things that are happening now, but I think it is quite clear that we need to have better ways of conducting these clinical trials. So how do we move things faster for the patients that we see in clinic? So the big thing that has happened over the past 20 years is that now we have a number of active research groups. We have a number of active patient organizations, and we all work quite closely together because it's quite clear that we need to have cohorts of patients that can be recruited into these clinical studies. And we also need to have much tighter link with industry because they are the ones that have the financial resources to conduct these initial phase 1 studies and the larger phase 3. Earlier in the year in February, we were all very fortunate, some of us, to take part in this scientific retreat, which brought together some of the most active groups and some of the finest brains in terms of determining how do we move this research further forward. And this is something which is quite important because research takes time, funding is limited, so we need to set priorities. And some of the priorities that we set for Leber's optic neuropathy, for example, is the fact that we need to collect more samples, we need to establish cohorts of patients that can be recruited into clinical trials. We need to have better models of the disease. And then we need to come up with a number of complementary therapies that could potentially rescue retinal ganglion cells and improve visual function. So one of the projects we're involved with here in the UK is to set up a tissue biobank from lung donors so that we can have more eyes and more brain that can be used by us and by other groups to try and better understand the sequence of events happening in the acute stage and the chronic stage as well. The one thing that I've kind of mentioned en passant is the fact that you get this selective vulnerability of retinal ganglion cells. But having said that, we don't have a proper explanation because the dogma has always been that you have a proportion of these retinal ganglion cells which are lost very quickly, and the reason for this is because they have a high metabolism, and therefore it's all down to ATP. But we know that this is quite simplistic because there are other cells within the retina, for example, photoreceptors, which have a much higher metabolic demands compared with retinal ganglion cells. And this is just a cartoon taken from a recent review suggesting that there are other things that can be involved in terms of compromising retinal ganglion cell function. You can have impaired mitochondrial DNA maintenance. You can have abnormal mitophagy, which is the removal of abnormal mitochondria. as we've seen for OPA1, you get an imbalance between diffusion and efficient. And I've not mentioned Wolfram syndrome, WFS1, and in this case here, you have a problem with the interaction between the endoplasmic reticulum and the mitochondrial network. And then the other thing that we also need to put in context, even if we put all these possible mechanisms together, none of them actually explain selective loss of retinal ganglion cells. And there are probably other neuroanatomical considerations here which explain why you get this preferential involvement of retinal ganglion cells. One of the excellent hypothesis put forward is that we lose retinal ganglion cells within the papillomacular bundle quite early because these axons are quite small and therefore they have reduced mitochondrial reserve. We also need to realize that myelination only happens behind the lamina clubosa, and therefore before that you get accumulation of mitochondria because you need more energy to be able to conduct the electrical signal. So it could also be that there is an anatomical choke point in terms of why retinal ganglion cells are affected in preference. And then the other thing is that retinal ganglion cells have very long axons which stretch up to the lateral genucleate nucleus. And the other thing that we need to perhaps discuss further is the fact that retinal ganglion cells do not exist in isolation. And if we look at, for example, these astrocytes, you can see a picture here taken from a review by the left, Jeff Reisman. You have these astrocytes which are surrounding the axons of retinal ganglion cells, and you can see these accumulation of mitochondria there. So it could be that not only is it about retinal ganglion cells, but the supporting cells are contributing to the early loss of these retinal ganglion cells. Finally, to be able to conduct research, not only do we need to have collaboration between clinicians and researchers, but obviously you need money because all of these things take time and we need to have the resources in place. So this is just a very exciting new development, at least in the UK, where LifeArc will be funding a number of centers, and one will be funded for rare mitochondrial disease under the leadership of Patrick Chinnery, Rita Horvath, and Mike Hanna, and Rob Taylor. And this is a very big program that will last for 5 years. And I'm very excited about this because this also includes a program for eye research, which will help us define better biomarkers and also move forward with clinical trials. So in summary, I hope I've convinced you that this is a very exciting phase for us clinical academics working in this field because there is already an approved treatment for Leber's optic neuropathy, IDEB and Raxone, but this is not a treatment that will be beneficial for everyone. Gene therapy, some very promising data, but also a lot of unanswered questions that we need to resolve. It's been slower for OPA1 dominant optic atrophy, but now certainly we're entering a very exciting translational phase. And both for nuclear genes and for mitochondrial gene, it is possible that gene editing will be something that can help us perhaps prevent the onset of visual loss in the future. And just a note that all these things that we want to achieve for rare disease is made more difficult by the fact that it's more challenging to recruit enough participants. And that's the reason why we need to have this much closer collaboration between centers across Europe and with other countries to be able to conduct these clinical studies as fast as possible. This is just a little picture here just to thank all the wonderful collaborators I've had the chance to work over the years, and we're still working very closely together, meeting on a regular basis. And I'm very happy to take any questions that you might have. Thank you.
Thank you very much, Patrick. Very interesting. Let's see if there is any questions in the chat. Let's see. I cannot see any questions. Maybe here. No.
I think there's one hand up from Anna Maria Sapupo.
I cannot see any from my view.
Okay. Do you have a question, Anna Maria?
Oh, yeah. Okay, now yes.
Yes, you can ask your question. Okay. I know that Anna Maria is having some problem. Yeah.
Let's see. Waiting for other questions, I can ask you something, Patrick. You mentioned,
Sure.
the 3460 mutation, the unusual behavior in relation to the ID-Benon therapy. Do you have any explanation for this, unusual, not beneficial effect of this therapy in this specific mutation?
For 3460?
Yeah.
Yeah. Yeah, I didn't mention this here for the benefit of time, but certainly, as you know, your group has published this paper on NQO1, suggesting that variants in NQO1 can modulate the response to idebenone, and this could be part of the explanation for this. I think the only way to be able to dissect that further is to look at a separate group of patients who are being treated with idebenone, where we've got the genetic profile, including NQO1, and try and determine whether the nuclear background might be influencing response. Yeah. So I think there's still a long way to go in terms of understanding why people respond and others are not, right? And it could very well be a good idea for future clinical trials, whether it's neuroprotective drugs, whether it's gene therapy, to potentially collect blood sample, before and after, to look at things as to whether or not responders are more likely to mount a mitochondrial bioenergetic response in terms of copy number. So I think we still need to determine that much more clearly, but with a very systematic plan about how we collect that kind of data. Yeah.
Yeah. So I can see a question from Valerio Carelli.
Hi, Valerio.
Is there any off target in your CRISP correction on OPA1?
Yeah, good question. It's certainly in the data that has been collected on these limited number of cell lines, there's no evidence of any major disturbance, right? But clearly, this will need to be investigated in other cell lines and also potentially, in animal models.
Yeah, so I cannot see other questions. So maybe it's interesting also to point out about, you mentioned about the labors with the very early onset, like before 12 years of age. So this is a very, special population.
Yeah.
So which is your advice about idebenone therapy for this specific population of patients?
Yeah. Again, I think this is something which is now very well reported, and with Piero Barboni, we basically put together this kind of big group of children who basically lost vision before the age of 12, both in the UK and in Italy. And it's quite clear that this is an unusual group where they behave differently, both in terms of how they present. It's not always a classical bilateral, sequential, bilateral simultaneous optic neuropathy. And also the response is different. You can have about a third of them who have spontaneous recovery and do pretty well, right? Now, the question about essentially, whether we should be treating or not is partly influenced by the fact that one is the EMA gave the approval for children, for individuals who are 12 years and older, and secondly, whether they have access to it in terms of the approval rules, right? Personally, if I have a child who's been affected with Leber's optic neuropathy at a young age, and we are able to provide idebenone, certainly I would recommend this, because there's no reason not to think that it will have a beneficial effect if someone responds. But as to how you do this, I think it's very much a variable. So I think I tend to be quite cautious. I would basically tell them to start at a low dose, something like 150 milligram rather than the full adult dose. And then we could check the bloods again in a couple of months' time and increase the dose to a high level, like 400 milligrams. But this is my kind of personal approach, and it is not evidence-based. So I don't know whether you have a different way of starting treatment for this subgroup, Chiara.
Yeah, so this is a very difficult group somehow, because as you mentioned, there is this spontaneous recovery. So this a good prognosis, even though you are not using the therapy. So without clear data, it's very difficult, but a cautious way of approaching, I think, is the best one. And also, I would suggest to check for the NQO1, as you mentioned, for the response can be influenced.
Yeah. And all of these things are quite important because just for the people in the audience who are not aware of it, but way back in I think 2016, There was this consensus meeting in Milan, and then the consensus guidelines for labels of the neuropathy was published in 2017. And we need to revise this because essentially a lot of things have come around the corner, including the LEROS study for the chronic group, whether or not there is a more problematic situation, the 346 mutation, including now the fact that NQ1 has been reported as a possible modifier of response. So I think all of these things are things that we need to review, hopefully sooner rather than later, because this gets quite complicated. The simple fact is, for example, for NQ1, this is not something that is routinely available in, I guess, most centers in the world, right? So even though some people might want to do it, they might not be able to have access to it until perhaps some commercial providers are able to provide this for people who do not have access to a local diagnostic service in their country. Yeah.
Yeah, I totally agree. Let me see if there is any other. This is the same. So there is maybe another question from Valerio. Later is better seems to apply also to Idibenon. Any thoughts on this?
Yeah, and I guess these are kind of observations, right? That certainly, Valerio, you're looking at groups of patients from different parts of the world, including the UK and Germany, and that seems to be the trend, right? And we also saw that for the gene therapy treatment, that you're not able to stop this catastrophic loss of retinal ganglion cells, at least in the first 6 months. And there's this observation that 6 to 12 months seem to be doing better in terms of the gene therapy window, right? Well, a lot of things have been put forward, isn't it, right? Could it be that if you're treating too early, you are faced with some kind of, barrier imposed by the swelling of the axonal segments within these retinal ganglion cells? But then, it's it's kind of counterintuitive, actually, really, right? So, I don't think I have any more, clear explanation for this, in the sense that, these are observations based on subgroup analysis, but perhaps we need to look into that more closely for future studies in terms of collecting blood samples, trying to profile their genetic background, trying to get more information on even the environmental background as well, right?
Yeah. Let me see. There is any other one? No, I cannot see any more.
I think one other thing, which I guess it's rather unfortunate that now the UK is outside the ERN network. But I think these are the things that we discussed, for example, at the London Scientific Retreat, and I think the same applies for OPA1. Is that increasingly there will be trials coming around the corner, whether it's drugs, whether it's gene therapy, whether it's gene editing. But I think going back to the basics, there's so much that we do not have available. For example, good prospective natural history data. Good data on, for example, what is influencing disease penetrance, why some people with long lose vision, others do not. Why is there such a big variability in terms of outcome for OPA1? Why do some people present very early with childhood development? I think it goes back to the fact that, at least, one of the things that the ERN network can put together is trying to assemble cohorts, trying to put together biobanks where we basically have enough blood samples, enough postmortem analysis, right? And I think that is something that could potentially arise from trying to bring together these centers through the ERN networks. Yeah.
Yeah, because as you mentioned, there are possibly many mechanisms behind the apoptotic signal which starts the disease. And if you are using just one of these approach, as you said, you are not able to change the natural history of the disease. So potentially we have to combine maybe different approaches targeting different mechanism of the disease, which is something that we can understand better maybe with the new models, IPs, or
So still a lot of things to be done now that we're 36 years since the mutation was reported in 1988. So we're making progress, but still very challenging for us, for clinicians seeing people in clinic. It's still a little bit disheartening that people are coming back for 6 month, 12 monthly review, and we still do not have anything tangible to offer. But I think there's a lot of positive things happening, and I think it's a combination of trying to understand the disease mechanism, but at the same time, we can't wait until we know everything. We need to go ahead with clinical trials, early phase clinical studies, as long as the preclinical data shows that the intervention is safe. Yeah.
Yeah. Okay, so we are 5 minutes ahead. If there are other comments or questions, otherwise we can just say hello to everybody.
Yeah, but again, thank you very much for inviting me, although I'm a non-EU country, really. Yeah. And I guess there are a few more activities happening this week and next week for the mitochondrial month, really, right?
Yes.
All right, Chiara, thank you very much. Bye to everyone.
Thank you, Patrick. Thank you, Chiara.
Okay,
bye-bye
bye.
to everybody. Bye. Bye.